Hello everyone. Our group has been working on antigen identification using randomized peptide libraries. My name is Tony Kawansi, and these are my partners.
I'm Joan Hart. I'm Tui Tran. I'm Kate.
Now, before we get into the nitty-gritty details about our project, there are some basics everyone here needs to know. These Y-shaped objects here are called antibodies, and they bind to an antigen at these binding sites up here. Now, an antigen is anything an antibody binds to, and it's usually a foreign invader, like bacteria, that make you sick. Now, once the antibody binds to this bacteria, the immune system comes and kicks it out of the body. As you can see in this depiction, this yellow antibody attaches itself to this bacteria, and big brother white blood cell there is going to go and kick him out of the body. Now, this system is supposed to keep you healthy, but it doesn't always. There are some diseases where the immune system actually attacks your body and makes you sick because it believes your healthy cells are the enemy, are invading bacteria. As you can see here, this healthy cell is about to get beat up by other healthy cells. Seems very confused. Now, we're not certified to work with blood. That's why we were in charge of looking for a model peptide sequence. But this model was designed to teach us the skills and procedures used by the other scientists in the lab we worked with. And they used these same skills and procedures to find the cause of autoimmune diseases, therefore helping prevent them and hopefully one day curing them. Now, my partner Tui is going to take you through generating a peptide library.
So in order to achieve the result we want for this project, we need to create a library of E. coli cells carrying the peptide sequences. In order to do that, first we need to insert a plasmid DNA inside a single E. coli cell. A plasmid DNA is a small circular DNA molecule within a cell that could replicate by itself. And the plasmid DNA works as an instructor for the E. coli cell to build the peptide sequences. Keep doing that, we will eventually create a peptide library that carrying the peptide sequencings right here. Using that peptide library, we could identify the reagent that the antibody binds to, and the scientists use that technique in the medical field to help preventing and treat diseases. So now Kate will explain for you how we use this library with the flow cytometry in finding the antigen.
Flow cytometry is a laser-based technology that is used in cell counting and cell sorting. We're using it in our project because it's good for the quantitative cell sorting. We start with a peptide library and then we bond antibodies to it. We're using antibodies that are bonded to a peptide and we tag them with fluorophore. After this happens, cells enter the flow cytometer where each individual cell is hit by a laser with a specific wavelength of light. The electronic signal is recorded by the computer. Cells can be separated by sorting or the information can be collected and analyzed. We were collecting cells that have high fluorescent concentration.
So this is the data we got from the flow cytometer machine for our 70 nanomolar sample, and the x-axis represents intensifying fluorescence and then the y-axis represents side scatter. And side scatter has something to do with the granularity of the cell. So since we're using mainly E. coli cells, they're concentrated in that tight little region right there. So primarily what we're focused on is the dots inside these boxed off areas because they represent the cells that are fluorescing above background levels. And we're interested in them because they have the peptides that are binded, or they have the peptides that the antibodies like to bind to. So in this first graph right here, it's the first sort which Kelly did for us before we came to the lab, and she separated out of the library population of 1 billion cells down to 30,000 cells. So we took those 30,000 cells and then we incubated them with antibodies and we did a second sort. And so you can see here that the the population of fluorescing cells is significantly higher than the first one, and we took it down from that 30,000 to about 3,000 cells. So then we went another time and repeated the process for another third sort, and then we got about 60% of the population to express these peptides and fluoresce. So pretty much with every sort that goes on, we're able to enrich the population with E. coli cells that have these expressed proteins that the antibodies would like to bind to.
After we sorted out the most highly fluorescent cell from the rest of the population, we extracted out the DNA from the cell and sent it for sequencing. However, due to time constraint of the internship, we were not able to perform the actual sequencing, so our mentor, Kelly, provided us 3 different amino acids motifs. So what are the amino acids? Amino acids are the small subunit of the large molecule protein. There are 20 different amino acids, so each of the amino acids changed right there. Each letter is the name of one amino acid. Using the 3 provided amino acids and put it into the protein database from ORICOM Plus, we were able to identify the molecule that the antibody binds to was actually the special cell receptor called HER2. So now John will explain for you what does HER2 mean and what does it do into the project.
So HER2 is normally found on breast cells, and then in normal amounts, they just help the cell grow and divide and repair with no problem. But in a certain percentage of breast cancer cases, there are way too many of these receptors, so they cause the cell to grow and divide uncontrollably, leading to a tumor. So we found out that Herceptin was the antibody that we were working with, and this works by binding to the HER2 receptor and preventing the signals that are telling them to grow. It just prevents them from receiving those signals. But it can also work by flagging down these cancerous cells and then alerting the body to remove them and destroy. And so thus shrinking the tumor and treating the breast cancer.
With the understanding of the connection between antibodies, antigens, and our immune system, using standard lab techniques and flow cytometry, we were able to sort and analyze cells from the E. coli library that represent bonding areas of antigens in order to create a library of cells displaying peptides that react with the antibody.
Using these cell libraries, we are able to determine what antigen causes the human body's immune system to attack itself or healthy cells and make patients sick. This technique has been used in medical research and has yielded great results in helping treat diseases. Finally, we'd like to thank the Condor project for allowing us this opportunity to work in the lab here at UCSB, the chemical engineering department for letting us use their equipment, and the Daugherty labs, but also our amazing mentor Kelly for teaching us all this. And we would also like to thank you, the audience, for listening.